anyone here who wasn't here last week, just to remind you that if you want to look at the revision notes, as Fiona calls them, they're on my website at that address. That's really the only thing you need to write down. Last week I reviewed what we did in our one session last year. I talked about the politics of revealing what you know about your ancestors to your DNA matches in order that you can work out how you're related to each other. And then I started to talk about how you have to fish in all the gene pools, with emphasis on the autosomal DNA gene pools. Your autosomal DNA is the DNA that comes 50% from your father, 50% from your mother, 25% from each grandparent. At the end of today's talk, I'm going to talk about Y-DNA, which only men have, and they get from their fathers, who got it from their father, who got it from their father, and so on, like your surname. So that is for surname studies. The biggest but not the best company for autosomal DNA is Ancestry DNA. The biggest and the best company for Y-DNA is Family Tree DNA. So if you're a male and you're interested in both, you have to spit for Ancestry and swab for Family Tree DNA. And Family Tree DNA will keep your swabs in storage, and they can do their own autosomal analysis on it. But if you want to be in all the gene pools, it's probably better start with Ancestry. You can download data from any of the companies that analyze your spit or your swab. You can upload to some of the companies, but not to Ancestry and not to 23andMe. So I went through a little bit of GEDmatch, a little bit of Family Tree DNA, and I think I said I would start tonight with Ancestry DNA. Somebody pointed out to me that Ancestry DNA have introduced a silly rule since I did mine, that every new kit you buy has to be associated with a different email address. But it's easy to set up a new email address if you want to get around that system. I did have one couple who were traumatized by this last year because the husband and wife have always used the same email address, and they ordered 2 Ancestry DNA kits and they could only register one of them. They couldn't figure out how to create a second email address. There are links here to the 3 main pages that you will need to use on the Ancestry DNA website. How many of you are already on Ancestry DNA? About half of you. One of the most important pages is the test settings page. There are my settings. Test details, name, now you know what year I was born, gender. The important one is the tree link. You can click there to change it. You need to have a family tree on the Ancestry.com website to link to your DNA. Mine is already linked into a tree which I call the direct ancestors of Paddy Waldron. It contains only my parents, grandparents, great-grandparents, and so on, none of the collateral relatives. If you're concerned about people hoovering up all your research work, you don't need to give them that information. You only need to show them the people from whom your DNA could have been inherited, who are your direct ancestors. The next thing down here, DNA matches. You can see your DNA matches and you're listed as a match. You probably want to leave that on. Your display name. I wish I could change the display name to my real name, but this kit is attached to my cousin's account because I've been doing this since Ancestry didn't want any DNA from outside the United States. So I used my cousin's address in Michigan, and henceforth I'm PW managed by C. Wood, and there's no way of changing that. Sharing preferences is very handy if you are working with a cousin and you want to see the cousin's full match list, or you want the cousin to see your full match list. You can just click change there. There's a list of all the people with whom I have shared my match list. I don't care who sees it. And it's often very helpful to see the match from both perspectives. And you might recognize some of the other person's matches that he or she doesn't recognize, and the other person might recognize some of yours that you don't recognize. It's a great help to be able to see it from both dimensions.
That would use from with 2 different emails on Ancestry.
Yes, you can have them both on the same account and share them that way. If they're set up separately, share each one with the other and use one account, pay the subscription on one account. Yeah. So that's the other thing. If you're managing kits for a dozen people, you don't need a dozen subscriptions. It is the subscription for the logged in kit that counts. So if the people that have shared their match list with me, I can see the family trees of all their matches through this account, even though they can't because they don't have the full subscription. So they're not quite as greedy as you make them out to be. You can get around the system somehow. And if you want to get your ancestry results onto GEDmatch and Family Tree DNA and MyHeritage and Living DNA, there's your download link to download the data file. And if you really decide to give up, you can delete everything, but don't do that, please. Then we have a match list. This is the top of my match list, where you can see my close relatives. They're in groups with labels at the top of them, and these labels are not labels from the English language. In this case, the first cousin label happens to be a real first cousin. The second cousin label includes 2 of my second cousins and a second cousin once removed. The third cousin label includes a half second cousin once removed or something. I think he's certainly not a full cousin. So these labels are not gospel.
And is that, did you create that?
No, Ancestry puts on those labels and you have no control over them. And even though it gives an exact label on the top, down here it gives a first to second cousin label, and a second to third cousin label, and a third to fourth cousin label. Those are obviously, because they include the full range of possibilities, more accurate than these very misleading labels at the top.
On Ancestry, I have 3 people who are down on Ancestry as third to fourth cousins, and actually all 3 are second cousins.
Yeah.
Yeah. But it's usually the other way around, isn't it?
Usually on Ancestry, they underestimate. No, Ancestry underestimates the closeness. Family Tree DNA tends to overestimate the closeness of the relationship. Here you can see how few people put up trees. There's one tree here. That's only there because I put it up for her, and it has figured out that we're relatives and given me a common ancestor hint. And over here on the right, there are stars, which are a built-in group. There's the full list of groups. I have decided the best way to use the star is when I work out exactly how I am related to one of my matches, I put it in the star group. You may have a different way of using it. Then there are up to 24 colored dots that you can use. I have decided to use the colored dots to indicate through which of my ancestors I'm related to this person. So because there are 24, I said that's 8 plus 16, so I created one group for each of my 8 great-grandparents and one group for each of my 16 great-great-grandparents. So because this is a first cousin, he is related to 8 of my 16 great-great-grandparents, half of them, all those on my mother's side, so I've given him 8 colored dots. These are second cousins. They're each related to 4 of my 16 great-great-grandparents, so I've given them 4 colored dots. This guy is a kind of a half cousin. My great-grandfather was married twice, and his 2 wives were first cousins. That's how he ends up with 3 dots. 1 for, 2 for the great-grandfather who we share, 1 for Michael McNamara, and 1 for Lucy O'Neill, who are our shared great-great-grandparents through having a common great-grandfather. And then there is 1 for Mary Galvin, who was the ancestor whose daughter and niece both married the same man. And then this one doesn't have a star because I haven't worked out, and I've marked her as an NPE because she's a very close match to my second cousin who doesn't know who she is. And there are people in that branch of the family with a record of having children out of wedlock, we'll say. So that's my way of using the dots. They're very powerful. None of the other DNA websites have them. And you can do nice things like show me all 6 people who are related to me through great-grandfather Waldron, and none of those is a confirmed relationship. What I have done is for each confirmed relation, so let's go back to a start match. And let's go down to one who I know exactly how she's related. This lady, when you click on the name, you get through to the individual match page, and then you can click on shared matches. And at the top, there are people I know, but this one I have given a colour dot for Anne O'Neill, one of my great-grandmothers, because I know she is a shared match with a cousin who's related to me through Anne O'Neill. So you can have a guess at who the unknown people are related to based on who the shared matches are. It would be nice if we had more than 24 of this group. So I'd like to have 2 for parents, 4 for grandparents, 8 for great-grandparents, 16 great-greats, and maybe even the next generation, where you've got to make do with the 14. And that makes it a little more complicated. So that's basically all I want to say about the match list. You can use those drop-downs, and there's a search button as well, and we looked at a match page. Quick look at my heritage. It's the newest of these websites. It has grown very rapidly. It can be quite slow. Here it is loading my 50 closest matches. It's got 2 copies of me because one didn't seem to be working, and now I'm afraid to delete them because I don't know what the rules are for how much you can see in terms of family trees and shared matches. Depending on when you upload it, there was a grace period when they started, and they gave people who uploaded early more features than people who uploaded more recently, and I can't get my head around the whole system. And I've uploaded some of my first cousins, and you can click there to review the DNA match, and I tend to open that in the new tab because it's so slow to load, and you can actually do that off the main page, and eventually it will load up shared ethnicities, and I think that will now disappear as it loads up other bits and pieces. It gets pushed down and it loads up shared DNA matches, and it shows the 10 closest matches shared by myself and my first cousin. Top one is my identical twin, and the next one is a known cousin, the next one is a known cousin, the next one don't know how we're related. But the nice thing here is you don't need to do all the sharing back and forth, because it shows the centimorgans shared on the left between myself and this person, and on the right the centimorgans shared by my first cousin, and this person happens to be my twin. So you'll very often see down there, you can see from the 920, that's a first cousin. Sometimes you see a mother, a child, a sibling, a close relationship. And you can put together the matches and family trees very easily, even though they haven't uploaded their family trees. And the annoying thing here is if I want to see who are my shared matches with J. Durkin, there's no open and new tab. I have to lose my place, and it replaces the shared matches with M. Durkin by showing me the shared matches with J. Durkin, and again, it takes forever to load up, and loads up the ethnicities before the shared matches. And I have tried uploading family trees for these people, but it says upgrade a subscription plan to add more people to your family tree, and I begrudge them the money. So it's getting there. When they started out, their matching algorithm wasn't working at all, and they were missing close relationships and predicting close relationships that didn't exist, but they sorted that out pretty quickly. So that is my heritage. And then there are other third-party tools and websites, which I'll give a quick run through. DNA Gedcom has one feature which I love, and it's called the Autosomal Data Segment Analyzer. And these were my matches at an early stage on chromosome 20. And I think this is a lovely graphical interpretation of exactly what's going on in the autosomal DNA matching process. I had about a dozen matches on this chromosome. I've taken out the names for privacy reasons. Probably should have done that with some of the other live demonstrations, but I think you get more by seeing the website live than you do by a screenshot sometimes. This one would be very, very slow to do live because it downloads a lot of information. It used to take 10 minutes to download, and now it takes about 10 hours to download the information, I think. So this first person matches me for 14 centimorgans at the start of chromosome 20, from 11,000 up to 5 million. Then there's a gap here where nobody matches me. then there's a whole bunch of people who match me on overlapping parts of chromosome 20. And then there's another gap where nobody matches me, and then there's a 7 centimorgan match at the extreme upper end of chromosome 20. But the nice thing about this group in the middle, it shows which of them match each other. So does the row match the column? Everybody along the diagonal matches themselves. The second guy is the pink, and also matches the third, fourth, and fifth people. The third guy in blue matches the fourth, fifth, sixth, seventh people, and so on. But there's these two down here who don't match anybody else. Well, what that means is you've got one group who are matching your paternal chromosome 20, and another group who are matching your maternal chromosome 20. And you can immediately see from the graphic in the middle, from the shared matches, who belongs to which group. What is more difficult is to tell whether the 2 are the paternal and the 8 are the maternal, or the other way around. So you would need some family tree information, or one of them to be a known relative, to separate them. All we can say at this stage is that the beige and the turquoise are on one side of the family, and the other 8 are from the opposite side of the family. So that's the first thing you might want to do with a new match, is see, is it on my father's side or on my mother's side? My first match was an adopted lady, I think I mentioned her the last day, who'd been trying for 80 years to figure out who her parents were. I couldn't tell whether she was on my father's side or my mother's side. My parents were both dead. My uncles and aunts were all dead. So I went to a first cousin on my father's side and a first cousin on my mother's side and said, please, can I have your DNA? And they were quite happy to oblige. And immediately I saw the adopted lady match the cousin on my mother's side, so I knew I was looking in Mayo for her roots, not in the parts of the country where my father's ancestors came from. So it is a great help to have at least one known relative on your father's side and at least one known relative on your mother's side. If you're lucky enough to have parents still alive, get their DNA as quick as you can, because you have only half of each parent's DNA and it's all mixed up together, and without these complications it's very hard to separate it out. So a lot of what you're going to be doing with autosomal DNA is dividing your matches into these, we call them triangulation groups, where you have 3 or more people who all match each other, and you can say which side of the family they're on, hopefully. If you're adopted, you've no hope. If you have known relatives in the system, it's easy to do. So if you want to run that outside, you go to the DNA GEDCOM website. You used to be able to do it on the website. Now you have to follow the links to download the client software. You can install the application for Windows or Mac, download, and it runs on your own computer. I'm not going to go through the details, and it can be very slow running. So sometimes you might want to leave it running overnight, but it's useful. DNA Painter is a relatively recent tool which people seem to love, again because it's graphical and colored. Here are my 23 pairs of chromosomes and the bits that I have been able to assign to particular ancestors. So that first black segment on my paternal side, again because my great-grandfather was married twice, I'm not sure is it on the McNamara side or the Galvin side. This green one over here I can certainly assign to great-grandfather George Clancy. Then on the bottom, the maternal side, again there are double relationships. In fact, I suspect this match is trebly related to me because of new discoveries recently, so I'm not sure which ancestor I got that segment from. That one I'm certain. That one I'm certain. So you can see down here I have colors for particular ancestors, and then where there's a double relationship I have an A or B type of ancestry. And the objective is to color in the whole of this chromosome map and be able to tell exactly from which ancestor which part of each chromosome came. And some people have done very well on it. It's a while now since I tried to update it, and I still have a lot of blanks. And that is called DNA Painter from dnapainter.com, where you also have an equivalent of that probability table that you were asking me about. But this is even easier. If you have a match with whom you share 123 centimorgans, you might want to know where might that match fit on the family tree. It's based on the same table that I put up earlier. So there's a 46% chance that 123 centimorgans is in the second cousin once removed category, which includes half second cousin and first cousin 3 times removed and other equivalent relationships. There's a 25% chance it's in the third cousin category, which is further out. There's a 12% chance that it's in the second cousin category, which is nearer, about the same chance that it's further out, more distant, third cousin once removed, and a small chance of getting that from a very distant match. So that's a quicker way than looking up the table. Some people prefer the table, some people prefer that. And the other nice tool that it has is what are the odds if you have a number of known relatives who you match and you don't know where you fit in the family tree. So this is very helpful for adoptees. You can set up a family tree like that and you can put in a hypothesis. So this person might be Daniel's son, might be Daniel's grandson, might be Daniel's full brother, might be Daniel's half brother, and it will tell you the likelihood of each. This one has a score of 0. It's just not possible. The purple dots, I think, mean a full relationship, and the yellow dots a half relationship. So this lady, he shares 760 with this lady. He couldn't be her uncle. He doesn't share enough to be a full uncle, but he shares enough to be a half uncle. So the yellow dots one is a possibility. He could be a first cousin, or he could be a first cousin once removed. And it gives the least likely possible outcome a score of 1. So the first cousin gets a score of 191. That means it's 191 times more likely that the person is a first cousin than a first cousin once removed of this lady Sissy. And there's no possibility at all of being a full uncle. A half uncle is equally likely with a first cousin. So it narrows down where the adoptee might fit on the family tree when you have 3 or 4 known relatives that all match the adoptee. It's very useful for adoptees and such like. If you're in all the DNA databases, you might want some sort of software where you can download all the information from all the websites and manage it together. A lot of people swear by Genome Make Pro. I have tried it, and the problem with these things is you have to keep downloading the new matches every so often. And I don't know, is it a lot easier to download the new matches every time and import them into desktop software, or just look at them on the main website? I tend to just look at them on the original website, but different people have different philosophies on that. Another thing that has come up recently is clustering. Grouping your matches into groups of people who are all related to each other, and those who all match each other are probably all from the same side of your family. Genetic Affairs is the state of the art for that, and it's also built into GEDmatch. So let me log into GEDmatch again, and it takes a while to run. I might leave it running and come back to it at the end of class. So I think that link may work. No, you have to go to it. Some of these applications you have to go directly from GEDmatch main menu. There's clusters at the bottom of the tier 1 menu, and it works that way. And if I put in my kit number, it starts building clusters, and it'll take a while. We'll come back to that later on when it's finished running. And while we're on third-party websites, I'm going to show you the big tree, which I love, which is for Y-DNA, not for autosomal DNA. So just a brief review of the tree of mankind, or the haplotree as it's called. This is what it looked like 20 years ago. Every man traces back to what we call the Y-Adam, not Adam from the Bible, but the only man who was alive in his day who has direct male line descendants living today. So there could have been thousands or millions of other men alive at the time, but this is estimated to be about 150,000 years ago, and he's the only one whose male line hasn't died out. So you can think that there might have been 1,000 surnames at that stage if they were using surnames, and only one has survived to the present day. And then there were mutations on the Y chromosome, and it divided into haplogroup A, and another group which subdivided into B, and another group which subdivided it into C, and they only went up to OR. So they didn't even need all the letters of the alphabet. And they thought these were the only types of Y chromosomes that would be identified. The science has come on enormously in the last 20 years since that simplified tree was put together. And now family tree DNA has a public version of the haplotree. Where there were 20 branches 20 years ago, there are now 20,000 branches. And some of those have several mutations on the branch that haven't been separated out. So across the top you have the original A up to OR, and eventually they added S and T, and then OR was divided into OR1 and OR2, and OR1 was divided into OR1A and OR1B, and then all those 1s and 2s and A's and B's got complicated, and they decided to give every new mutation a label like M173. So there's 11,000 men in the family tree DNA database in that M173 branch, and 10,000 of them are in that M343 branch, and 10,000 are in that L754 branch, and so on down along. And you get over here on the right the countries that they say their most distant known ancestors came from. And it's not the best way to look at it. I much prefer the big tree version. That haplogroup OR counts for about 80 to 90% of all men in Ireland. So these top level letters are more like continental level haplogroups. And the O's and the I's are very common in Europe. So now there is a thing called the big tree, which overlays the surnames on the haplotree. So up at the top we have all the mutations. This is the branch starting from P312, which again is probably a quarter of all men in Ireland will be somewhere on this branch, and you can follow the tree down by clicking on a leaf, and that blows up that leaf, and down at the bottom of the leaf you have the surnames of all the people in these branches of the tree. So you can see these mutations only occur in men with the surname called Crozier, but then there are mutations where there's no consistency in surname, but you have Horde and Hoare, and maybe the surname mutated and looks a bit different today. These mutations only occur in men called Duncan, but some of them spell it with a C and some with a K. And you can scroll over to the right. And you mightn't want to look at the whole thing together, but you can look at any small branch as well. You can click on that branch and you can say, show me the block tree, and it just shows you the two Weirs. Again, the DNA is the same, but the spelling has mutated. One is Weir with an A, the other is Weir with an I. And we were talking about Griffins and Griffiths earlier. I should have looked for the Griffin and Griffith branch, but I didn't think of doing that. I belong to this U106 branch, which is kind of a shadow branch. You can't get to it from the main website. Here's my tiny little sub-branch. I'm the only Waldron. I have no Y chromosome match who has the surname Waldron. These matches probably have a most recent common male line ancestor with me before the use of surnames at all. I don't think Wade is a variant of Waldron. Maybe it is. Cheney certainly isn't. These mutations only occur in Beatties. I may have a common ancestor 1,000 or 1,500 years ago with the Beatties. Or there may have been a surname DNA switch. A Waldron may have become a Beattie, or a Beattie may have become a Waldron. But I have no immediate evidence of that. I have a wonderful Waldron family tree of a family that came to Ireland with the Ulster plantation in 1609, and the surname has daughtered out. So I cannot find another Waldron of that family tree to compare my Y chromosome with. Eventually I might find one, but if it really has daughtered out, I'm not going to find him. If you want to be on that big tree and you have done Y-DNA analysis with family tree DNA, it's like copying your autosomal DNA data to GEDmatch. You put it in the Y-DNA warehouse and you have the links there to get into the forum to tell you how to download the file and upload it to the warehouse, and then they'll add you to the big tree. If you're not from haplogroup OR, I don't think there's any equivalent. The other haplogroups are not as far advanced, but if you're an Irish male, there's a good chance you are from haplogroup OR. How are we doing for time? God, we're nearly out of time. I was going to talk about shared matches and triangulation, but I think I will skip over that for now and go on to the rest of the Y-DNA stuff, because I want to talk to you about surname projects, something I've had questions about. The great thing about family tree DNA is that it runs these projects. They originally started out when Y-DNA was the only game in town as surname projects. There are now also geographical projects. Here is the Griffin surname project. I'm not sure quite what they call it, but they've accounted for all possible surname spellings in the title of the project. It has 704 members. Each project has a home page with various sections, and the one I tend to go straight for is the DNA results. You can donate money to the project. It can only be used to purchase DNA tests from family tree DNA. So you might club together and get one member of the family to swab. There's no point in having three brothers all have their Y chromosome analysed because they all have exactly the same Y chromosome. Better get the top of the range analysis for one than the cheap analysis for all three. You have links there to the administrators. I hate the way, if you're a project administrator, anybody in the whole internet can see your email address and then spam you with requests to get your 3.5 billion out of Nigeria or whatever. The spam is usually about. It's probably racist to say that, but that's what a lot of the spam emails are about. So what you get on the DNA results page, you get a colorized chart in which the project administrators can group the project members together in whatever way they like, but typically the closely related members grouped closely together. So Griffin is a very common surname. Griffith is a very common surname. Griffin is probably more common in Ireland. Griffith or Griffiths in Wales. God knows what's more common around the world. So these are divided up into which of those ancient top-level letter haplogroups they belong to. There's some Griffins from haplogroup E who are all considered to be closely related. First column is the kit number. The second column is the surname. You can see Griffith, Griffiths, Griffin, all in the same group. This column is very important for helping with genealogy to fill in the name of the most distant known male line ancestor. Most people have done it, but there's a few blanks. This column shows you in red where you're predicted to lie on that modern 20,000 mutation haplotree. You can buy two types of Y-DNA test. The old one was based on Str mutations. There are all those columns over here to the right. The modern ones are based on Snp mutations. The Snp mutation is a mutation that happens once in the history of mankind. So one man has this mutation. At his time, nobody else has it, but all his sons and their sons and their sons and their sons have it. And that's how they can be put together in a family tree. So the green ones are the ones who've done the more modern testing. The red ones are the ones that have done the older testing. The values of the Str mutations are numbers, the number of times a particular string of letters is repeated on the Y chromosome. And if there's a difference from the mode within the group, it's highlighted. So here's a group with only two people, and they've exactly the same Str mutations. There's no colored highlights. And if we scroll down, here's a good group from haplogroup G, Griffins from haplogroup G, where they mostly have the same Str mutations, but here some have a 17, some have an 18. There one man has a 28, everyone else has a 29. But they're so similar, you say they're related within the surname era at least. So there's Griffins in haplogroup E, haplogroup G, haplogroup G, haplogroup I, lots in haplogroup I, a few in haplogroup J, haplogroup N, and then lots and lots in haplogroup OR down at the bottom. So there's one group here with a lot of greens, and they're County Kerry Griffins, and they've all done the advanced recent SNP testing. And you can see the pattern over here. They all match apart from one or two. And the early mutations, then there's a division between 25 and 26, which works exactly with the 31 to 30 division on that marker. And you can actually, within the last couple of 100 years, work out, in some cases, where this mutation occurred, what house it occurred in, even in one case, I know. So if you're a Griffith or a Griffin and you join this project, the project administrator will figure out where you belong. Are you a haplogroup O Griffin or a haplogroup I Griffin? Are you a Kerry Griffin or a Clare Griffin or a Tipperary Griffin? Wonderful things can be done with it. Now my clusters are ready. So it has put my matches together. They are currently sorted by the average amount that I share with the other people in the cluster. I don't know who most of these people are, but they're all related, closely related to each other and to me. Here's my fourth cousin, and I don't know who these two are, but they also match my fourth cousin who has duplicate kits. And you really need a very large screen. There's a nice big group, but I don't know how they're related to me. Here's a nice group of about 5 in turquoise. All 5 of them are known relatives to me, and I can understand why they're all together. If I had as good a family tree on my mother's side as I have on my father's side, I'd probably be able to identify where the other groups come from. Problem was, my father's father wrote down the family history at a very early stage. My mother knew nothing about her family history. Nobody had ever written anything down, and the church records in Mayo don't go back as far as they do in Clare and Limerick and other parts of the country. So you get a lot of nice artwork out of this, as well as genealogical information. Let's look at a couple of other projects. I run the Clare Roots project for anybody with ancestry from County Clare. Homepage is the same sort of thing. I have grouped people on the DNA results page. I have a co-administrator over in America, and I've written up a little bit about the objectives and how to join. I've also put together a surname list. So they're all the ancestral surnames of the people with Clare ancestry who have joined the project. They have asked me on the activity feed page, can you please add my surname to the group? Hopefully they're mostly Clare surnames, and it tells you how many people in the project have that surname in their family tree. So there's 24 project members out of 1,300 with Blake as an ancestral surname, 28 with Barry as an ancestral surname. And that can hopefully be some help. Project administrators can also do nice things with autosomal matches, but there's no real public display of those. And the activity feed is for people to discuss with each other and with the project administrator. You have to be logged in to see it. And it's a bit like a Facebook group for discussing County Clare ancestry. And I guess in this day and age, a lot of those sort of discussions tend to happen in the Facebook groups rather than in the DNA projects. Come on, log in. We don't really need to see what the activity feed looks like. It's like a Facebook group. Then when you figure out where you belong on the haplotree, you can join a haplogroup-based project. We have 2 big branches of the Tree of Mankind in Ireland that you might have heard of. The Nile of the Nine Hostages branch, very prominent in the northwest, distinguished by the M222 mutation. It has its own project. L226 is the Dalgashian branch. The Dalgash were the ancient people of County Clare and the surrounding area. Brian Boru was probably the best known member of the Dalgash. So that's actually the O'Brien fairly modern ancestral home in Dromolan Castle at the top of the L226 website. And there's a fantastic O'Brien surname project. And Conor O'Brien, the present Baron in Chiquin, has been a very enthusiastic participant in that project from a very early day, and he has an unbroken, well-documented 30-generation pedigree going back to Brian Boru, and in the 1800s his title almost went extinct and it passed to an 8th cousin, and there was a very long procedure to get the title, the 8th cousin, recognized as the heir to the title. So they were wonderful pedigree, and we can actually date the DNA mutations for the O'Briens to particular people on that 30-generation pedigree. and they're all in this general Dalcashian project as well. Make sure if you want to appear on those results pages to set your preferences on that page and on that page, or if you don't want to appear, do the same. If there isn't a project for your surname, set up your own. There's a link to the application process. And I've got far too much material on here. Some of the things that the project administrator can do for you. Hopefully I have the right link there. On those STR mutations, if you pay for Y37 analysis, you're deemed to be a match if there's 4 or fewer differences out of the 37 markers. If you pay for 67 markers, you're deemed to be a match if 7 or less of those 67 are different. If you pay for 111, if 10 or less of the 111 are different, you're deemed to be a match. So sometimes you can have somebody just outside that threshold. It's a fairly arbitrary threshold with the same surname with a known pedigree. So here was a guy who I think he had only one match, and we didn't know where he belonged to, but he's done 37 markers. There's one guy within 4 out of 37. There's 3 others within 7 out of 37. They're all from the M222, the Nile of the Nine Hostages branch. So I could immediately tell as project administrator that he must be from the Nile of the Nine Hostages lineage. He couldn't tell that from his matches within genetic distance 4 out of 37. I could tell it from his matches within genetic distance 7 out of 37. And you can, if you set up your own project, you can recruit in various different ways. You might have an offline one-name study or clan association or something, sign them all up. Family Tree DNA will, if you set up a project, send out an email on your behalf to all of their customers with the relevant surname, encouraging them to join your project. Or you can look at the matches of anyone who's in the project and say, there's another Clancy, or there's another OD, or there's another Griffin. I'll write to them myself and invite them to join the project. And that way you build up a critical mass of people with the same surname and work out how they fit together. I run the OD project and the Clancy project. We actually worked out that half of the ODs in Ireland seem to really be O'Briens. At some point in the Middle Ages, the O'Briens and the ODs used to foster each other's children, and at some stage there was a surname DNA switch, and an O'Brien child with O'Brien DNA was fostered by an OD family and adopted the OD surname. So we call them the Royal ODs. They go right back to Brian Boru. And then there's the real ODs. We're also Dalcashian, but the common ancestor is before Brian Boru and before the use of surnames. The same thing in the Clancy project. Back to the 1200s and 1300s, there were paper records of Clancy's near Doolin in northwest Clare, and records of Clancy's near Chadaree, Newmarket and Fergus in southeast Clare. And it was always assumed they were of the same lineage, but in fact there are 2 Clancy lineages in County Clare, different DNA signatures. And I could make a pretty good guess at which one is the Doolin branch and which one is the Newmarket and Fergus branch. But I haven't quite proved it yet, because they moved all around the county, around the country, around the world, and where they live today is not indicative of where their ancestors lived 800 years ago. That's another major caveat with the use of DNA. You hear people say in the media that DNA tells us about human migration. It does not. If person A matches person B, yes, they had a common ancestor, person C, who may have lived where person A lives today, or may have lived where person B lives today, or may have lived in some completely different place. You need a lot of other information added to the DNA to build the migration patterns that the genetic anthropologists or population geneticists or whatever talk about. It's not as simple as DNA telling you where your ancestor lived. It can only tell you where your relatives live, unless you can actually go and dig up an ancestor who lived 1,000 years ago. And that is now being done. And here in Trinity, in the genetics department, there is a very good ancient DNA lab run by Dan Bradley. And the major problem they face is there was about 1,000 years of Irish history where cremation was all the rage, and there were no remains to dig up. So from about 1000 BC backwards, and from the Christian era about 400 AD forwards, they have lots of remains to work on and can use the present day DNA and the ancient DNA to build family trees, but there's that 1000-year gap with all the cremations, and they're desperately trying to figure out how do we get DNA out of cremated remains, and they haven't figured it out yet. Anyway, I'm out of time. I could talk about this for weeks. This is your last session of the term, and Fiona will be back to you in January, and hopefully I've inspired you to do a little more with your DNA. I have a couple of more family tree DNA kits if anyone's interested. As I say, Ancestry DNA has the biggest database for autosomal. Family tree DNA is the best for Y-DNA. Any questions? If you have a train to run for it. If you haven't, I can answer your questions. So thank you all for listening, and I hope you've learned something.